kindly confirm the presence. अगर मेरी आवाज़ पहुंच रही है तो कंफर्म करें. Because the icon does not represent the connection of the students, so it is necessary to give message. अगर मेरी आवाज़ पहुंच रही है तो कंफर्म कर दें. Kindly give message. आज ये आप लोगों को बायोकेमिस्ट्री का द टॉपिक इज रिलेटेड विद द बायोकेमिस्ट्री एंड दैट इज द ट्राई कार्बोक्सिलिक एसिड साइकिल एंड इट इज पॉपुलरली नोन एज क्रेब साइकिल. एंड नाउ वी आर गोइंग टू डिस्कस on the Krebs cycle. Initially, in the when we discussed the glycolysis, at that time I told you that the end product of the glycolysis is pyruvic acid, and that pyruvic acid is converted into the acetyl coenzyme A, and that acetyl coenzyme A enters in a cyclic breakdown of the 3 carbon molecules, that is the tricarboxylic acid. And the one initially in the glycolysis we observed that one molecule of glucose is responsible for production of 2 molecules of pyruvic acid. That is, the glucose has 6 carbon, and the 6 carbon glucose breaks into 2 molecules of pyruvic acid. Each has the 3 carbon, and that the 3 carbon pyruvic acid is converted into the acetyl coenzyme A in the presence of oxygen. When the process is going to complete by the aerobic respiration, then the pyruvic acid is converted into the acetyl coenzyme A. In one way, it is the pathway of the glycolysis, and it is necessary to remember that the acetyl coenzyme A is actually responsible for the initiation of Krebs cycle or activation of the Krebs cycle, popularly known as the Krebs cycle. Actually, it is the tricarboxylic acid cycle, and the tricarboxylic acid cycle is completed in the 8 steps, which we are discussed today. That what is the 8 steps, which enzymes are associated, which coenzymes are associated, and the net product of the Krebs cycle is in the form of carbon dioxide and reduced form of the NADH and FAD. So it is the, if you consider the acetyl coenzyme A, just I am telling that actually the acetyl coenzyme A is responsible for breakdown. Is responsible for initiation of the Krebs cycle. And acetyl coenzyme A, in one way, it comes from the breakdown of glucose, and in another way, the acetyl coenzyme A is synthesized by the breakdown of the fatty acids, and popularly known as the beta oxidation of the fatty acids, which is also recommended in the syllabus of the MSc and NET examination, which we will discuss later on. But initially, it is necessary to remember that the acetyl coenzyme A is formed from the pyruvic acid, which is synthesized by the breakdown of glucose in one way. In another way, it is synthesized from the beta oxidation. Beta means the two carbon, alpha and beta carbon are oxidized to form the acetyl coenzyme A in the metabolic or in the catabolic actions of the fatty acids. So the first step, I am just saying, you remember that is the two parts. One, that is the conversion of the pyruvic acid into the acetyl coenzyme A, and the acetyl coenzyme A is responsible for the initiation of Krebs cycle. So the I am going to explain the complete part in two heads. One, the initiation of the Krebs cycle, which is associated by the action of citrate synthetase enzyme and responsible for the synthesis of citric acid from the oxaloacetic acid. It is the first step of the Krebs cycle. The first step of the Krebs cycle or the initiation of the Krebs cycle is associated with the or is initiated by the conversion of oxaloacetic acid into the citric acid in the presence of acetyl coenzyme A. That is the cyclic pathway, and it is completed in the 8 steps, in the 8 reactions. So the first, we are going to discuss the conversion of pyruvic acid into the acetyl coenzyme A. Then the pyruvic acid, if we study simply in any book, then you all very well known that the pyruvic acid is responsible for the synthesis of acetyl coenzyme A, and in this process the coenzyme A is associated, and one molecules of NAD is reduced into the NADH plus NADH and H plus, and one molecules of carbon dioxide is eliminated. So when anyone asked, or in your question if it is asked that what is the number of carbon of glucose enters in the Krebs cycle, then the answer is only 2, not the 3, because one molecules of the carbon is eliminated in the form of carbon dioxide when pyruvic acid is synthesized to acetyl coenzyme A, and acetyl coenzyme A enters in the Krebs cycle. So only 2 carbon of glucose enters in the Krebs cycle. It is the simple way, it is a simple line, but it is not simple as mentioned in one step, because here the enzyme associated is the pyruvate dehydrogenase, because here the H plus is released, so the enzyme is dehydrogenase group, and it is associated with the pyruvate dehydrogenase because it acts on the pyruvic acid. But it is not simple, it is a complex process, and the enzyme pyruvate dehydrogenase is a multi-step thing enzyme because it is a complex, and the in the pyruvic acid synthetase, the pyruvate is a 3 carbon compound, just I am saying, and it releases 1 molecules of carbon dioxide and forms a 2 carbon compound called acetyl coenzyme A. That acetyl coenzyme A enters in the Krebs cycle. In this process, a carbon dioxide is released And here, so the dehydrogenation and decarboxylation, because the carbon dioxide is released, so the decarboxylation and dehydrogenation, because one H plus is released, NADH plus H plus, and one molecule of carbon dioxide is released. So both process, the decarboxylation and dehydrogenation, are involved in this process. Though the biochemical reaction is catalyzed by enzyme pyruvate dehydrogenase complex enzyme, because it is completed in the three steps. First, and some important facts which is necessary to remember, that the enzyme, first point to remember, that TPP or thymine pyrophosphate, FAD, or flavine adenine dinucleotide, and lipoate are very much essential for this reaction, as these substances help in the oxidation of pyruvate to acetyl coenzyme A. It is an irreversible reaction. Here it is the irreversible reaction. Second point to remember, that in this reaction NAD plus is oxidized to NADH and H plus. Now the pyruvate dehydrogenase enzyme induces three enzymes. Just I am saying that it is a complex process, and here the pyruvate dehydrogenase induces three enzymes, and these enzymes are dihydrolipoyl transacetylase, second dihydrolipoyl dehydrogenase, and third pyruvate dehydrogenase, or the same enzyme is sometimes referred as pyruvate decarboxylase. So these three enzymes are related with the coenzyme A synthesis, and the reaction which is responsible for the synthesis of acetyl coenzyme A from pyruvic acid. The fourth one, which is remember, that this reaction takes place in mitochondria. Inside the mitochondria, this reaction takes place in mitochondria. Fifth one, that is important, that I am just saying that it is the main part or main component which is responsible for the activation of Krebs cycle. Now the coenzyme A has two carbon from the glucose. Sorry, acetyl coenzyme A has the two carbon from the glucose, and the coenzyme A, if we consider the coenzyme A, then the oxidized two carbon molecules which is now entered in the acetyl coenzyme in the acetyl group of the coenzyme A is attached with the coenzyme A, and the coenzyme A is an organic molecule. It is an organic molecule derived from the vitamin B5 to form acetyl the steps of the acetyl coenzyme A. In the first step, the pyruvate is released to carbon dioxide, and the TPP action, just I told what is the full form of TPP just earlier, and it converts into the acetyl TPP complex, and this step is catalyzed by enzyme pyruvate dehydrogenase. And now the pyruvate dehydrogenase action, after the completion of the pyruvate dehydrogenase action, the acetyl TPP complex is associated with lip, sulfated lip, and form the lip means lipid, lipoyl group, lipoate group or lipoyl group, and it converted into acetyl lipoate, and then the TPP is released. The acetyl group is associated with the lipoate, sulfated lipoate, and it is converted into the acetyl lipoate, and now the TPP is released. Here, the enzyme involved is dihydrolipoamide acetyl transferase, because it is responsible for the transfer of acetyl group. So it is referred as the acyl group, not acyl group. Then it is referred to the acyl transferase, and then acyl lipoate is converted into acetyl coenzyme A. After the association with coenzyme A component, then the acyl lipoate is converted into FADH, and then the associated with the FAD and converted into FADH, it is the enzyme which is involved here, the dihydrolipoyl dehydrogenase. So here the three components are associated, three enzymes are associated. One, pyruvate dehydrogenase; second, dihydrolipomide acyl transferase; and third, dihydrolipoyl dehydrogenase. In this process, the FADH transferred the H plus to NADH. So here the NADH, one important and significant information we obtained from this discussion, that the NADH is not directly converted into the NADH plus H plus, but the H plus is at first associated with FAD and then converted into the FADH, and then the FADH donated the H plus to the NADH, and the NAD plus H plus is released. So here the one molecules of CO2 and one NADH plus H plus are released, and the glucose, pyruvic acid is converted into the acetyl coenzyme A, and this acetyl coenzyme A enters in the Krebs cycle. So when we study that the pyruvic acid is simply converted into the acetyl coenzyme A and releases the NADH plus H plus and carbon dioxide, that is the dehydrogenation. The enzyme, the step is associated with the dehydrogenation and decarboxylation. Actually, it is a complex structure in which the pyruvic acid is converted into the acetyl coenzyme A. And here the two molecules of carbon of glucose is present in the acetyl coenzyme A. Then the acetyl coenzyme A is associated with the oxaloacetic acid, and it converts into the citric acid, and the oxaloacetic acid, the citric acid forms the three carbons, two carbons of the acetyl coenzyme A and one carbon derived from the oxaloacetic acid. So it converted into the citric acid, and the enzyme which is involved here is the citric acid synthetase, and coenzyme A is released again in the system. Now the citric acid is initiated the Krebs cycle, and it is the first step of the Krebs cycle. So the Krebs cycle is also referred as the citric acid cycle because it is the first molecule which is responsible for the initiation of a cyclic breakdown of tricarboxylic acids and release the carbon dioxide and hydrogen. So here we easily understand that two carbons of the glucose enters in the Krebs cycle. It means the two carbons enters must be released in the form of the carbon dioxide by the Krebs cycle. So here one thing again clear that the two carbon of the glucose is converted into the carbon dioxide in the Krebs cycle, whereas one carbon is converted into the carbon dioxide at the level of the acetyl coenzyme A synthesis. So I am giving all the informations or knowledge thread wide because sometimes the questions are asked in your net examinations and they are the simple questions, but because due to the lack of the proper understanding, the students give wrong reply in the examinations. So it is necessary to understand all the concepts here. Now the citric acid is converted into aconitic acid, and the aconitase is responsible for the reorganization, intermolecular reorganization of the carbon, hydrogen and oxygen, and the citric acid is converted into the isocitric acid. It is actually the reorganizations of the molecules, and so the citric acid is converted into isocitric acid, and the enzyme which is involved here is the enzyme aconitase, because the aconitic acid is an intermediate product between the citric acid and isocitric acid, or in other way it is responsible for the aconitic acid is an intermediate which is responsible for the reorganization of the molecules of intra-reorganization of molecules of the citric acid. Now the isocitric acid is converted into alpha ketogluteric acid, and in this stage it is the dehydrogenation. In the dehydrogenation, so the enzyme which is involved here is the isocitrate dehydrogenase. It is the third step. First is the synthesis of citric acid. Second is the reorganization method that is responsible for the reorganization of the citric acid to the isocitric acid. And at the third stage, the isocitric acid is responsible for the synthesis of alpha ketogluteric acid. And it is the dehydrogenation, and the enzyme is the dehydrogenase. And on the basis of the substrate, the enzyme is named as isocitrate dehydrogenase. So during the dehydrogenation, NADH and H plus are released, and when the NADH and H plus are released, so one molecule of carbon is also released in the form of the carbon dioxide to uh, rearrange the molecules, and now it forms alpha ketogluteric acid. That means at the alpha carbon, ketone functional group is ketone functional group is attached, and at the fourth step, the alpha ketogluteric acid is again dehydrogenases, and here it is a complex process. It is a complex process where coenzyme A is associated in the reorganization, and the enzyme is alpha ketoglutarate dehydrogenase, and that reorganization and involvement of the coenzyme A here, the one molecules of NAD plus is converted into NADH plus H plus, and when it is organized, the coenzyme ASH is attached by the, and in this process one molecule of carbon dioxide is also released, and the component formed is the succinyl coenzyme A, and that the succinyl coenzyme A is an unstable molecule. It is not stable, and due to the energetic instability, the succinyl coenzyme A is reorganized, and in this reorganization, the succinyl coenzyme A synthetase is involved, and here the one molecule of GTP is synthesized at molecular level or at the substrate level. It is not through the oxidative phosphorylation, but at the substrate level, one molecule of GDP is synthesized and it converts into the GTP. So it is again an important point here. It is here in books, the ATP is mentioned. Of course, it is not wrong, but the ATP obtain the high energy phosphate group from the GTP, not directly the ATP is synthesized. So the succinyl coenzyme A, when converted into the succinic acid, the one molecule of GTP is synthesized, and that GTP transfer the high energy acyl bond or high energy acyl group to the ATP. So one molecule of ATP is synthesized, and coenzyme A is released in the system. Now the succinic acid is formed. It is the fifth stage. And now in the sixth step, the succinic acid is converted into fumaric acid. The succinic acid is converted into fumaric acid, and here the enzyme dehydrogenase is FAD linked, not NAD, one FAD. And here the FAD is linked. That FAD is converted into FADH2, and fumaric acid is synthesized at the sixth step. Now the fumaric acid is reorganized to malic acid. It is reorganized or formed, the fumaric acid is reorganized to malic acid, and the enzyme which is involved here is the fumarase. In this step, one H2O, one water molecule is involved in the conversion of fumaric acid to malic acid. And now the malic acid is again converted into oxaloacetic acid, from where the citric acid is synthesized, and then the malic acid is converted into the oxaloacetic acid in the presence of enzyme NAD-linked dehydrogenase. And now the NAD is converted into NADH plus H plus, and oxaloacetic acid is formed. That oxaloacetic is responsible for again initiation of the Krebs cycle and synthesis of the citric acid when acetyl coenzyme A enters in the mitochondrial matrix. So, in the complete Krebs cycle, three molecules of NADH and one molecule, sorry, three molecules of NADH and one molecules of FAD and two molecules of carbon dioxide are released, whereas in the step of the pyruvic acid to acetyl coenzyme A, one molecules of carbon dioxide and one molecules of NADH are released. So now it is a cyclic stage, and which is responsible for the synthesis of a reduced NADH and FAD, then NADH and FAD carried to the F1 particle, base of the F1 particle, where they are responsible for the transfer of H plus either to the coenzyme Q or FAD, flavin-linked FAD, at the electron transport system, and responsible for the initiation of the changes of potential, H plus potential, and releases the energy to the system, responsible for the synthesis of ATP. And this step we have already discussed in my last class when I discussed the electron transport system and changes in the redox potential. Now we discussed today on the Krebs cycle and associated with the oxidative respiration, and it is your present in your syllabus. Now you all, if you have any problem, you give me message, and I will give you the reply. For if you have any problem, anyone has any problem, kindly give me message. I am waiting here for 1 minute. Probably, as I think, now I am going to close my live session. Probably you have no any problem, and you must prepare for the facts related with the Krebs cycle. Thank you. Thank you to all for your patience hearing. Thank you.